doing at this point. And I think we're all hopeful that something positive will come out of that. But we clearly all have a very difficult task in the next decade in terms of maintaining food resilience and productivity without causing more problems. So I think legumes are a critical part of that. Thank you.
Thank you, Sean. So at this point, I should also thank the local organizing committee from ICRISAT, represented by Dr. Arvind Kumar, the Deputy Director General for Research. Then Dr. Sean Mays, the Research Program Director of Accelerated Crop Improvement. And from Indian Council of Agricultural Research, Dr. Sanjeev Gupta, who is the Deputy ADG of Oilseeds and Pulses. And the guidance from our Director General, Dr. Jacqueline Hughes. So now I request Jackie to give her welcome message and also ICRISAT at 50. Over to you, Jackie.
Thank you, Janila. So good morning, good afternoon, and good evening to all of you. And those of you who are still a bit asleep, please wake up. It's my pleasure to welcome you to the 10th International Conference on Legume Genomics and Genetics. And as you heard, this year also marks our 50th anniversary, 5 decades of service to the smallholder farmers of sub-Saharan Africa and Asia. ICRISAT works on the resilient, the climate smart, the nutritious legumes and the cereal crops, chickpea, pigeon pea, groundnut, sorghum, and millets, including pearl millet and finger millet, which are critical to the agri-food systems of the drylands and for nutrition globally. As you will know, and you'll hear more about, our core strength is that world-class research covering areas ranging from the genomics, gene editing, modern crop breeding, natural resource management, seed systems, nutrition, and climate smart agriculture. On the other hand, to sustainable agribusiness models, digital solutions, and the policy inputs. And all of our research interventions are anchored in a value chain approach, which spans the whole production system and the food value chain from input to consumer, as well as all the associated ecosystems that support the food value chains, including policy support. As I was thinking about what you're going to be talking about, I wanted to ground it a little bit more in why we're doing all of this. And ICRISAT is uniquely positioned to develop solutions that contribute to achieving the Sustainable Development Goals. And that's by focusing on the drylands, which are increasing, improving food production and the nutrition security, because the drylands are most vulnerable to climate change and environmental degradation, and they are growing. The crops that I mentioned, our mandate crops, which includes, of course, the legumes, have a great potential to contribute to the sustainable, nutritious agri-food systems under this changing climate scenario. And perhaps more importantly, to serve as a risk mitigation measure, particularly for nutrition, for resource-poor smallholder farmers. We implemented with our partners the Tropical Legumes Project in 13 countries of Africa to do that very thing, increase productivity. And that was through improved varieties and quality seeds. For that work, ICRISAT was awarded the Africa Food Prize 2021. We also provide technical support to the Pulses mission of the Government of India, which has enabled India to become self-sufficient in pulse production. ICRISAT, we contribute to the agricultural research systems across Africa and India by developing the genomic resources in chickpea, pigeon pea, and our other crops. The most important resources are the genome assemblies, the sequence data on thousands of lines, the gene catalogs, molecular markers, high, mid, low density genotyping arrays. And these are being used for trait discovery and deployment in crop improvement programs across Africa and Asia. With our partners, ICRISAT has mapped several economically important traits in our mandate crops. more than 50 in chickpea and more than 20 in pigeon pea. And these high value traits are critical for national crop improvement programs. To do all of this, we need to remember that ICRISAT maintains the world's largest gene bank for millets and legume germplasm accessions, 130,000 accessions in long-term storage. And specifically for this meeting, there's 20,000 accessions of chickpea and about 13,000 accessions of pigeon pea. The subsets of the germplasm diversity, the core and mini-core collections, have served as a gateway to access and use the diversity for trait development and crop improvement in many national programs. More than 1,100 or so high-yielding, climate-resilient nutrition varieties of the legumes and cereals have been released by partners in different countries from the breeding lines developed by ICRISAT. And these include 55 varieties of chickpea and 54 of pigeon pea, which contribute enormously to increased productivity. For a specific impact, work with our partners has led to the chickpea revolution in Andhra, Telangana region of India, where ICRISAT is based, where production was increased 8-fold in 15 years. And the chickpea varieties developed by ICRISAT were instrumental in scripting that success. These new varieties cover over 90% of the chickpea area in Andhra, Telangana. We've also shared over 160,000 seed samples of many breeding lines, and they've been shared with agricultural universities and research institutes. To change a little bit more, to again focus on why we do all this, youth engagement, gender, social inclusion are a cornerstone of our research agenda. We always adopt a gender dimension in our research, and this contributes to sustainable and appropriate development pathways in the drylands. We're all after creating impact. And our research around gender goes beyond collecting and analyzing sex disaggregated data. It goes further. We examine whether the interventions, including new varieties of these legumes, have actually addressed gender barriers, accommodated gender differences to improve technology uptake, and therefore the welfare outcomes. We look at those gender implications around these new research products for policy, programs, and institutions. And for us, the approach is to be gender aware, gender sensitive, and gender transformative. By doing so, we provide that essential diversity context to research in the drylands and critical support to research strategy development, priority setting, research utilization. Some of you may be thinking this commentary is a little out of place because this is a conference on legume genomics and genetics. But I believe that those gender dimensions are especially critical here, as in many other areas that we work. So the research arena, to make sure that women have a greater input into research, but also to focus on the needs of both women and men. This year marks the 200th birth anniversary of Gregor Johann Mendel. Janila mentioned it, and Noel will be speaking in a minute. And he laid the mathematical foundation for the science of genetics. For me, it's more than fitting that he experimented with pea, a legume crop. And the other point is his experimentation came from his training. I'm sure we'll hear more about that, but physics and mathematics. And in that same vein, I urge you to think outside the boundaries of our own, including my own, very narrow discipline, if we are going to revolutionize the field of genomics. And I'd like to congratulate the International Advisory Board and the organizing committee for developing this splendid online conference. It's always better to have a conference, however short it is. And it's an impressive lineup of speakers and young researchers. I wish you all success and look forward to the outcomes. Thank you.
Thank you, Jackie. Thanks a lot for that motivating welcome message. And as you mentioned, as ICRSAT realizes its vision and mission goal of delivering nutritious agri-food systems, I think this platform of engaging with the basic science researchers and applied research would be the best fitting as we work towards that goal. Thank you for that. And yeah, I mean, you touched upon the gender lens, gender being aware, sensitive, and I forgot that. I'm sorry, but anyway. So, I mean, for this conference, the IAB has evaluated the abstracts received from different researchers and young professionals, and we selected 6 because we had 6 slots for this, and then we just wanted to see the gender, just in case we are not having at least 50% of the representation of women, and then we were surprised to see all the selected presentations of young professionals were women. So that's one. So and now may I call upon Noel Ellis to present the honorary talk on this ICLGG on Mendel at 200. Before that, I would like to introduce Noel to the, many of you in this group know him, but then I would like to do a formal introduction of Noel. Currently, Noel Ellis is holding a part-time position in legume biology at John Hunts Institute, UK, and he's working closely with research projects of Kathy Martin on grass pea and with Claire on a pea program. So Noel had his honors degree in molecular biology and his PhD from University of Edinburgh. And in 1979, he moved to John Hunts Institute, where he focused on pea genetics and genomics. And in 2011, he was in the University of Aberystwyth as professor of crop genetics. And in 2013, he was here with us at ICRSAT as the director of CGIAR's research program on grain legumes. And during 2015 and 19, he was holding a position of honorary professorship at University of Auckland, and then returned to Norwich in 2019. So over to you, Noel. Next 30 minutes is your time. Then I would like to remind the participants to drop in their questions in the chat box. And at this point, I would also like to thank Jackie and the other leadership team members who have supported the hosting this program. And there is a message in the chat box that this conference is live on YouTube as well. The link has been shared. So over to you, Noel.
Okay, I hope that you folks will play the video. If you don't, I can do it from here. Otherwise, I'll just turn off my video and mute at the moment.
The video will be played from here, Noel.
Good, if you can play it, great.
So a little bit about what we know about each of the characters that Mendel studied.
Roop Kiran, may I just interrupt? The sound quality, we are not able to hear. It's very poor. May I just interrupt? The sound quality, we are not able to hear. It's very poor.
Yeah, madam. One moment, madam. We are sharing from another computer.
Janilla, if it's the recording itself, then I can take over and try and do it manually.
Sure, Noel.
Just let me know how you would like me to proceed.
So, Rupkiran, do you want a minute to try from the other system, or should
Yes, madam. Give me 1 minute time. Madam.
Okay, okay, thanks.
Yes.
And I should point out that when Mendel described... Good morning. I'd like to thank the ICLGG committee for giving me this chance to speak to you on the occasion of the 200th celebration of Mendel's birth, and to describe to you some of the genetics of these amazing characters that Mendel studied in his 1866 paper. So I'll say a little bit about what we know about each of the characters that Mendel studied. I should point out that when Mendel described these, as is shown over here, he always described the characters in terms of a difference between the two forms. And it's important to realize that at the time that Mendel was discussing these, was writing and was studying these things, it was not really known how meiosis worked. Meiosis was unknown, and even the process of fertilization was not very clear. So it was difficult to understand what the relationship might be between a seed on a plant and the plant on which that seed is formed, and where the independence of a seed comes from. Is that different between when it's still attached to the mother plant, or does it happen after it's sown in the ground? So there are two types of characters. The first pair of characters that Mendel described were what we can call seed characters. So, for example, round versus wrinkled seeds, or green versus yellow cotyledons. And the other characters were characters that are expressed in the mature plant, i.e. after the seed has been sown and the plant is growing. So it's important for Mendel to study both of these things. And once I've discussed what we know about these various characters, I want to discuss some issues that arise from Mendel's paper and have been discussed elsewhere. Johann Mendel was born in 1822 in the village of Hinchinsey in Silesia, now in the Czech Republic, possibly even in this very room. He grew up there, was educated locally, and eventually moved to the monastery of St. Thomas's in Brno, where he took the name Gregor. And he intended to become a teacher and part of the teaching role of that monastery. He wasn't entirely successful at this, but remained a teacher at the Realschule in Brno for many years, where he taught natural sciences. As part of his teaching activities, he was for a while in Vienna, studying at the university, and where he studied natural sciences. And on return from Vienna, he undertook a series of experiments in the breeding of vegetable crops. And this lasted for a period of about 8 years until 1865.
So
During that time,
let me interrupt
he
again.
progressed from working with vegetable breeding, particularly breeding peas, which he described as one of the most nutritious crops, to try to understand the mechanism of inheritance, answering the Abbot Knapp's question, what is inherited and how? Mendel's work on pea genetics and breeding ended when he gave his talks to the Natural Sciences Society in 1865, and then in 1866 his presentation was published as a paper in the proceedings of that society under the title of Experiments on Plant Hybrids. In 1867, Mendel became the abbot when Knapp had died. He continued to do a number of biological experiments, including his work with Hericium, which was eventually published in 1870. But thereafter, his biological experiments on plants, at least, came to an end, and he continued as the abbot until he died in 1884. At that time, Mendel's time, his work was not really widely appreciated in the scientific community. It wasn't until De Vries, Correns and Chermak published their papers in 1900 that Mendel's work on genetics came to the fore. And it was in the 20th century that genetics really developed into a science in its own right. And it had a number of controversies which I'll not discuss, although there were a number of criticisms raised against Mendel during that time, and I will talk about those later. Genomics and genomic tools.
Noel, you said that you would be able to do live, so maybe that may be more engaging. So can you come in live?
Okay, I'll try and do that. Can you hear me?
Yes, we can hear you.
Okay, and I'll try. Oh, yes, I have to share screen. So can you see my screen? I seem to have lost it.
We are
Yes,
seeing
we
your
can see
Zoom
your
screen.
screen.
We have to see the
Okay.
presentation screen.
So you can see me,
Yes, and the Zoom screen.
and I can't see the Zoom.
So you can close this Zoom window and then go to the, yeah, you can go to your slide, to Mendel at 200, and then you can click that, whichever.
So you can see my screen at the moment, can you?
Yes, we can see your desktop screen, yes. So
Excellent, good.
you can,
So,
you can see the slides now. Thank you, Nora.
okay, so I'll start at a little way into it because we've lost some time. And So I just given you a little bit of information about the history of Mendel, where he was born, and the history of the behind his breeding program, and the sort of 8 years of work that led up to the publication of his paper in 1866 after his two talks to the Natural Sciences Society. I just want to point out that Mendel continued working on plant biology after those papers, but on a slightly different topic with Hyraceum, shown in this picture of the plant. And that was after he became abbot. But as we know, his work wasn't really appreciated at the time. And I then said a little bit about the history of genetics in the last century, but pointed out that it wasn't until the 1900s that people started to characterize Mendel's genes. So I want to talk very quickly about those, and then maybe say a few words about Mendel's paper itself. So the first of the genes to be described was round versus wrinkled, and this is a consequence of defect in starch biosynthesis. This was shown by Madan Bhattacharya and colleagues at the John Innes Center in 1990. It's sort of prehistory to most people, I suppose, but it was really a combination of biochemical techniques that allowed Alison Smith and colleagues to show that the defect in the wrinkled line was a defect in an enzyme called starch branching enzyme. So Madan with Kathy Martin and so on were able to isolate the gene using antibodies and an expression vector. And then I was involved in the genetics, which showed that there was a difference in restriction fragment length polymorphism, if anybody can remember what that was, that co-segregated with round and wrinkled. And then Madan showed that the reason for this was that there is an insertion in the 3' end of the gene close to the end, but nonetheless resulting in a defective product. And that insertion, interestingly, was an insertion of a small transposon, a DS-like element, and that has since then it's been shown that there are a range of several different genes which, when mutant, give you the same phenotype, and all of these genes are defective in various parts of the starch biosynthesis pathway, but it's only this gene R, that encodes the starch branching enzyme, that was available as a mutant form in Mendel's time. So we know that that's the gene he worked with, and we know from work in Claire Dominy's lab that the insertion in the 3' end is the only one that we can find in the extensive P collection at the John Innes, the only wrinkled mutant allele at this locus. Though I noticed that in the recent new genome assembly, Yang et al. say they found a different one, but I think we need to investigate that a little bit further to understand exactly what's happening there. I think some sort of allelism test between what they've described as a dimpled form and this needs to be done. The second of the genes that Mendel described was the difference in cotyledon color. He called it endosperm, but it's not the endosperm, it's the cotyledon. And you can see it's either yellow or green. This is a picture from Claire. It's in this case, the recessive form is the green form and the dominant form is the yellow form. And a couple of papers in 2007 showed that, in fact, what we have here is a mutation in a gene that at the time was called stay green, but later was shown to be a defect in a gene called the magnesium D-chelatase. So you can see why the dominant type is the yellow type. It's able to degrade chlorophyll, whereas the mutant is not able to degrade chlorophyll. So the third gene that Mendel discussed is, or I should say, he didn't discuss it as a gene, he discussed it as a character difference between white flowers and colored flowers, though interestingly, he referred to it as the seed coat color. The seed coat is a maternal tissue, so this does not segregate on the F1, on the seeds on the F1 plant, because the test is a maternal tissue. But it's very interesting that he described it the transition between talking about seed characters, where the difference is seen on the F1 plant, before he started talking about the mature plant characters. White flowers were known in agriculture for a long time. The earliest reference we could find was from Pietro di Crescenzi. He wrote a monograph on agriculture in about 1300. It's a handwritten copy. It's not really properly dated, but about 1300. There are versions of it that were printed later that you can see, but we know that it's been in agriculture for a long time, so presumably it's quite an old allele. Not like this is in contrast to both R and I. In R, like A, which is flower color, there is really just the one allele. There is a second little a allele, but very, very, very rare. But for cotyledon color, there are multiple alleles. So that's a very unusual state of affairs for a recently arising mutation. Okay, so in 2010, Roger Helens and other colleagues worked on this gene, the sort of little consortium that we had, and showed that the nature of the defect in the white flowered mutant is a splice site, donor site mutation, which leads to an extra 8 nucleotides from the intron occurring in the transcript. So it creates a few additional amino acids and an end-frame stop. So that was clearly why this gene is defective. And the other very minor allele we found in a small number of accessions also it has a frame shift mutation. So the fourth character that Mendel described was what he called a difference in the form of the ripe pods. And this is the presence or absence of a thickening layer of cells, which you can see in this orange podded type, where there's some pigment in that tissue. So when this is present, the pod wall is stiff and it has this inflated phenotype. And if this is absent, the pod wall is weak and it is collapsed around the seed. So if the seeds are nice and sweet, and this is pod walls collapsed, these are nice to eat in a stir-fried or as a vegetable. So that has been known also for a long time. Recently, Zhang et al. Produced a very nice paper which had some GWAS analysis, and they identified 2 peaks corresponding to the presence or absence of this pod wall thickening, and these probably correspond to the genes P and V in P, which are duplicate genes that give rise to this mutant form, and they suggested that it may be encode a pectate lyase. The fifth of the genes that Mendel described was the difference in pod color between green pods and yellow pods. Incidentally, he referred in passing to an additional variant, which is actually a common type in a traditional pea cultivation, particularly in the Netherlands, where you have these purple podded types with a dark colored testa called a capuchin pea. Okay, so that's this form with this beautiful purple color. If you like, that's an eighth of the character number 8 from Mendel. Okay, so this green versus yellow pod has the opposite type of dominance from green versus yellow in the cotyledon. Here, the green type is dominant and the yellow is recessive. Interestingly, this shows that the nature of dominance isn't a consequence of the type of the phenotype, it's a property of the gene itself. A couple of years ago, Shirazawa et al. Did an analysis in F2 population and showed that a gene encoding a 3' exonuclease co-segregates with this phenotype in their F2 population. But as with the other GWAS, it's not a demonstration that that's what the gene is. It's just a candidate and a tightly linked candidate. The sixth character that Mendel described was fasciation, and he described this as the position of the flowers on the stem. And you can see why that is, because here we have a fasciated mutant with the flowers bunched at the top of a fasciated stem. This is in contrast to wild-type forms where the flowers form at successive nodes. Again, this has been known for a long time. Here's a nice picture of Pisum umbulatum from Gerard's Herbal in 1597. Pisum umbulatum is how Mendel also referred to this character. So again, a trait that was deployed in agriculture. So the final character that Mendel described is what he called plant height. And he measured this in non-metric units. I don't actually know whether these are the same feet and inches that we have in the USA and then in the UK to some degree. But anyway, there's a clear difference between the tall and the short, as is measured by the plant height. But this difference isn't a consequence of the number of nodes, it's a consequence of the distance between the nodes. The tall forms have a longer internode than the shorter internode. And there are several of these types of dwarfs known, but the one available to Mendel is this one, LE, which encodes an enzyme involved in the maturation of gibberellic acid from, it converts GA20 to GA1 or GA9 to GA4. The common variant of this is a missense mutation, which has a dramatic effect on the enzyme activity. So it's not a complete null. Okay, so I'd like to say a few words about a ninth character that Jim Weller attracted my attention to, that Mendel discussed towards the end of his 1866 paper, where he said that he did a cross to examine the genetics of flowering time. And in this case, he said the flowering time of the hybrid stands almost exactly between, or almost exactly in the middle between the two parents. So this is clearly a case of semi-dominance. And we know that the gene LF in P, when mutant, has a semi-dominant effect on flowering time. So Mendel said he was going to do some experiments on this, but in a letter that he wrote to Nagley in the end of 1866, or maybe it was the second letter in 1867, I can't quite remember, he said he wasn't able to pursue these experiments because of the bruchid infestations that he had in Bernal at the time. Presumably the bruchid population had grown very extensive because of his 8 years of work with huge numbers of plants. So he had to abandon this experiment. However, there was a paper published in 1915 from Hoshino, who was working in Japan. And these are experiments that he started in maybe 1908, so not very long after the so-called rediscovery of Mendel's paper. And here I've just redrawn one of his tables where he's tabulated days to flowering, and he's put in the boxes the number of different plants which flowered on those days. And I've colored them white or purple according to whether they were white flowered or purple flowered. A day that's colored blue has both white and purple colored flowers occurring. So you can see that the parents are quite distinct in their flowering time. And in the F2, when he planted the F2 families, the white flowered types generally flowered early, and the late flowering ones were generally colored. So if you like, one of your earliest examples of a QTL or maybe even a GWAS study. And we know that this is because the gene LF is tightly linked to the flower color gene A. So Mendel was studying, in fact, both of these characters. So it's very interesting that had it not been for the Bruchids, Mendel would have pursued 3 subjects that are not generally associated with Mendelian genetics. That's to say, semi-dominance, linkage, and quantitative characters. Okay, so I want to change my tack a little bit here and talk about some of the criticisms that have been leveled at Mendel's work. And these are basically, what I'm going to discuss is not really what I would call the Albion view, but to do with what Fisher and other statisticians had to say about Mendel's paper. So Fisher made some really quite horrendous claims about Mendel and led to a myth that Mendel had done something funny with his data. So I'm going to explain what this was. And basically, Mendel did an experiment to look at the number of types that you would see in the dominant class of an F2. He had shown that the F2 segregated 3-to-1 for the dominant and the recessive class. He had a particulate theory of inheritance that explained why this was, why you would have twice as many that did not breed true as would breed true in this dominant class. So he did this experiment with the seed characters for which each F2 plant had a lot of F3 seeds, so he was able to get a lot of good data on the frequency of heterozygotes in the F2s for those characters, showed that it was a very good fit to his experiment for the or his theoretical prediction of a 2-to-1 ratio. And then he did it for all of the whole plant characters. And I've illustrated that here in with respect to pod color. So what he did was he selected 100 F2 individuals of a particular class and grew an F3 family from each, and looked to see whether the recessive character segregated in the F3. If it did, then it came from what we would call a heterozygote. If it didn't, it came from what we would call a homozygote. And Mendel got a very good fit to the 2-to-1 ratio. Fisher and others, it has to be said, others later, pointed out that you would expect occasionally that the F3 families would be mistakenly classed as a dominant class, because just by chance, if you like, the 11th pod wasn't seen. So you should have a little bit of misclassification of heterozygotes to homozygotes. And Fisher worked out what that expectation would be. So we can compare what these expectations are. So what I've plotted here is the frequency distribution of the number of dominant F2s in 100 individuals expected based on the 1-to-2 or 2-to-1 ratio, or the ratio that Fisher calculated. And I've circled Mendel's data in red, so you can see that Mendel's data are distributed around the peak of the 2-to-1. ratio, but a little bit, there's a sort of an excess on one side of the peak in Fisher's ratio. So slightly biased ratio given Fisher's expectation. And it is this criticism that has led to a lot of things said about Mendel's data. But I should point out two things. First of all, Mendel repeated the experiment with the green versus yellow pods. This was the first experiment, this is the second experiment. So the second data point is dependent on the first data point. In other words, if the first data point didn't have this value, Mendel wouldn't have repeated the experiment. So you have to bear that in mind. The second problem is that we see that parchmentless and tall and dwarf are very similar to each other, more similar to each other than the two GP experiments. And we know from Mendel's letters and also from the report from Eichling that there was linkage between, there was a cross in which Mendel had both these characters segregating. There was a four-factor cross, in fact. And we know that V and LE are linked. And in this cross, they would have been in repulsion phase. So you wouldn't have seen something funny in terms of the frequency of double mutant phenotypes. So it's possible that the data for the P and V and LE are not independent if some of them came from the same F2 individuals. So in other words, there are probably two fewer points on this side of the peak than expected. So there are some reasons to think that this is not the case. Another issue is whether, in fact, there was exactly a 2-to-1 ratio in this cross. And if you look at you can work out what the allele frequencies were in these individual populations from the F2 data that Mendel presented, and you can see there is actually a very slight bias in the allele frequency, as there always is in a cross. But this bias is in the direction in favor of the interpretation that Mendel gave, rather than Fisher's. Data that was available to Fisher, I have to say. Finally, there's been some criticism about Mendel's data as a whole. Mendel presented a huge amount of data in tabulated form, and sometimes gave examples of individual classes in those forms. And so there are a number of different ways you can aggregate the data. So what I've done here is plotted the frequency distribution of chi-square values that Mendel found that can be found in Mendel's data in a variety of different ways of aggregating it. And I've given the standard deviations of the chi-square values in those classes. And you can see that Mendel's data points are generally within the standard error bars, as you would expect, except for these four examples with large chi-square values, which are in fact four values that Mendel pointed out as being unusual. There are illustrations of how far the data could be from the prediction. So as a conclusion, I'd say there's nothing unusual in Mendel's data whatsoever, apart from the clarity with which he presented his results and his willingness to present data that could be taken to challenge the data. So with that, I just want to thank thank Madan for sending this nice picture of Kathy and Alison and me from 1990s. Lots of people who worked on the A gene, and Peter van Dijk, Julie, and Martin Swain, with whom we worked together on these statistical analyses. And finally, just to thank the funding agencies. Basically, that's BBSRC, who funded the John Innes Centre, funded me my salary for many years, including at the moment. Also, DEFRA, who funded a lot of work at the John Innes, and the EU, who have been very supportive of P genetics. I should say also we have some support from DEFRA, which helps me to work with Noam Chart in the genome genetic resources unit at John Innes, but not a subject I'm talking about today. So I'll stop sharing my slides and maybe answer some questions. I hope you were able to hear me. My apologies if it was my problem with the recording.
Thank you, Noel. The presentation was clear. We could hear all the slides. Fantastic. So from Mendel's discovery, and now we know what the gene is responsible for that phenotype which Mendel has seen. It's nice. And then when we have asked the participant to post their questions in the chat box, but as you know, in the program, we will be taking these questions towards the end of the session. So now maybe it's time for Michael to come in from University of Queensland and make an advertisement for ICLGG 2023. Thanks a lot, Noel. Enjoyed the presentation thoroughly. Yeah.
Thank you, Noel. Hello, everyone. Welcome from Brisbane, Australia. My name is Michael Advardi. I'm at the University of Queensland. All right, I'm going to share my screen. Oh, sorry. Okay. Janelle, you still hear me? You're muted. Okay. Give me the thumbs up. Can you hear me? Great. All right.
Go ahead, Michael. We can see the screen.
Yeah, it's not the one I want to give you though. I want to change the view. Just trying to work out how to do that. You're seeing the preview screen here, is that correct?
Michael, you could go to the display because you may be using two screens. This is Karamboard Sadiq here.
Yeah.
You may have a laptop and a screen. So go to the display setting and then swap.
Yeah, I'm trying to find that actually.
Yeah, I can see it. I can see it here. So go to your laptop.
Yeah.
And then go to the click display setting, and then
where is that?
Which? Oh, that's on your laptop. I can see here on the top, but it may be on your laptop. Icon click.
Yeah.
So if you go to your laptop, yeah, and then display setting up the top of your toolbar.
Yeah, I'm looking for that, but it's kind of, oh, here it is. Yeah, sorry.
Good, it's good now.
Yeah, hang on. How's that? All right.
No, it's gone back now. Earlier it was good when you give, but it's okay. It's okay. We can see it.
All right. Okay. Well, welcome again, and gee, I've got. You can see pictures here.
Yes, beautiful pictures we can see.
Okay. Well, that's yeah. Let me see display settings. Are you seeing the full screen?
Not the full. Now we are full. The full screen is visible. Yeah.
Okay. All right. So yeah. Well, we will host the ICLGG in 2024 in Brisbane. Why Australia? Well, as you probably know, we're a home to world-leading research in plant science, agriculture, and food. There's an opportunity for this conference to align with a conference called TropAg, which is the largest tropical international conference in the world. It had 1,000 people just recently here, and it focuses on everything from basic biology through plant breeding, agronomy, and business related to tropical agriculture. So there's a great opportunity for us to get a bigger view of the role of legumes in agricultural systems, especially tropical systems. So that's going to be held in parallel with our meeting. We have a bunch of passionate local organisers, including Brett Ferguson, Eloise Fu, Peter Gresshoff, April Haswell, Nijat Imin, Willie Mathesius, Celine Menz, myself, and Jim Weller, and others. So you will be in good hands here. Brisbane is a subtropical climate. It's a safe, compact city, world-class infrastructure, friendly, and it will be hosting the 2032 Olympic and Paralympic Games. So you'll get a taste of Brisbane before all of that, I guess. It's easily accessible. We have more domestic flights than any other city in Australia. And we have a second runway that will deliver flight capacity on par with Hong Kong and Singapore. There's over 20,000 rooms of accommodation and over 10,000 within a walk of the convention centre, with a range of accommodation types, including budget options. The conference centre is on the south bank of Brisbane, south of the river. It has a world-class convention infrastructure. It's on 16 hectares of parkland, with river walks, rainforest pockets, and a swimming beach, with over 90 bars, cafes, restaurants for you to enjoy. If you've got an Australian bucket list, you'll be able to tick off a lot of the things from Brisbane. We're centrally located to a lot of the best things in Australia. This should be a video. So let me see if this works. It should have sound, but apparently the sound isn't working. It's just a music background. But while we go through, I can give you a couple of my renditions of Australian birds if you like. So here's one. That's a kookaburra. And at this time of year, we've just been bombed by magpies, and they have an interesting sound. It's something like this. Anyway, otherwise, the place is fairly quiet, as you can see on the video, but the infrastructure is great for a conference like this. Anyway, let's see. Anyway, so thank you for your attention, and we look forward to welcoming you all here in 2024. Thank you, Janella.
Thanks, Michael. That was nice. Nice. So this is the 2023 call advertisement for ICLGG. I think all the participants have made a note of it, and the location as well, and of course the science quality.
Great.
Thanks, Michael. So we now move on to the next presenter for the day, Sonali Rai from Tennessee State University, USA. Sonali, let me introduce you to the group. Some of them may know you from your work on Rhizobium symbiosis. Sonali Roy has her MS from Bose Institute, Kolkata, India, and she has her PhD from John Innes Center, Norwich. She worked on a thesis titled Analysis of Gene Expression, Regulation and Function of 3 Symbiotic ABC Subfamily B transporters in Medicago truncanulata. So she's been working in the area of rhizobium and symbiotic nitrogen fixation since her PhD. That's what I can see. And she was a postdoctoral fellow at Nobel Institute in USA. And now she is a faculty assistant professor at the Tennessee State University. And I mean, one of her paper in Trends in Plant Science, I think that was well read and well cited by all the researchers in the legume community. Over to you, Sonali. Look forward to hear from you on genetic discoveries in legume nodulation.
Yeah, thank you so much, Janella, for the introduction and for inviting me. Can everybody hear me? I hope so. Okay, so I'm going to start. Yeah, okay, thank you. I'd just like to say that I really enjoyed the last talk. Thank you for that, Noel. Yes, I learned a lot. So my name is Sonali Roy, and as of 2021, I'm an assistant professor at the Tennessee State University in Nashville, Tennessee. And as the title of my talk today says, I'm going to be speaking about genetic discoveries in legume nodulation and symbiotic nitrogen fixation. So I'd like to begin by thanking the organizers and the International Advisory Board for inviting me and letting me present at the 10th International Conference on Legume Genomics and Genetics. And the only regret I have is that I'm not able to attend in person in Hyderabad at ICLGG. And a lot of it has to do with the amazing food that Hyderabad has that I've heard so much about from my students and while I was in India. And so it's really fitting that the International Conference on Legume Genomics and Genetics is held in Hyderabad, because Hyderabad has this rich diversity of legume-based food that it offers. And this includes the Hyderabadi Halim, which is made with chana dal. Then there's the Hyderabadi Khatti dal and the Hyderabadi Khichdi, which is made with masoor dal, which is lens. But it also includes some more unique foods, such as the Bobbari Garelu and the peanut-based Andhra Bengan. And so all this delicious protein-rich plant-based food is part of our rich culture and heritage, and that we are so proud of. But it's not true for the rest of the world, which still relies on animal-based meat as its primary source of protein, which is not so great for the environment. And so I was discussing this in 2020 in the middle of the pandemic with Michael Udvardi. And so we were discussing over email, how do we ensure sustainable access to protein-rich food for 7 billion people and counting? So he replied, and I quote, so he said, we need to eat more vegetarian food and follow in the footsteps of 1 billion Indians. And so while this might have been a little bit biased, because one of his favorite cuisines is Indian food, as is mine, he is of course correct. And we know that legumes are a major part of this, because if you look at starch-rich tubers such as taro, you get less than 3% of the daily protein requirements. Barley and other cereals are more nutritious, but you could get almost 40% of your dietary protein needs with legumes such as chickpea. We know, and this audience, I really don't need to tell this audience, is that legumes are a part of sustainable agriculture. They are the cornerstone of a sustainable agriculture, and they provide solutions to the dual problem of nitrogen use in agriculture. So what is the dual problem of nitrogen use in agriculture? We know that nitrogen is a limiting nutrient for production in natural ecosystems. And as this image clearly shows, on the right, you have plants which have not been supplemented with nitrogen, and they are stunted and yellowing. But if you add just a little bit of nitrogen-based fertilizers to the plants, as on the left, they are dark green, they are taller, they are flowering, and they look healthy. So this boost in plant productivity upon addition of just a little bit of nitrogenous fertilizer is what fueled the green revolution of the 1960s. And countries which could afford these expensive fossil fuel-based nitrogenous fertilizers did really well, and they met their food security goals. But over time, what happened is that we started using so much of this nitrogenous-based fertilizer that in 2014, the United Nations declared excess nitrogen in the environment to be one of the 5 major threats to our planet. Countries everywhere had to spend billions and billions of dollars to try and mitigate the effects that it had on human health, on the ecosystem and the environment around us, and the climate as well. Much of the nitrogen that is in our environment is from agricultural runoff. Because if you think about it, we've really amped up the amount of nitrogen that we provide in soils, which are from fertilizers, from manure, and their biologically fixed nitrogen. But plants cannot and do not take up all of it. Almost 50% of the nitrogen that we supply as fertilizers is not taken up by the plant roots, and they are lost to the environment as runoff or as gaseous emissions. So the dual problem of nitrogen use in agriculture is this: if you use too little nitrogenous fertilizer, you're not going to meet the population, world population's food demands. But if you use too much of it, it's detrimental to the environment. It's a cost to the farmers. And because it is fossil fuel based, its use is unsustainable long term. So as plant biologists, we need to think about how can we decrease agricultural dependence on nitrogenous fertilizers without losing the benefits of extra nitrogen on plant growth. Legumes, of course, do this naturally. We know that legumes can independently can fix nitrogen independent of any added fertilizer. And they do this by associating with soil bacteria or beneficial soil bacteria called rhizobia, which are able to convert this abundant nitrogen, which is in our environment. 70% or more of it is nitrogen. But plants, just like us, are unable to use it unless it's provided in a form that they are able to absorb. So these bacteria convert atmospheric nitrogen into a form such as ammonia, which the plants are then able to take up. So this process is called root nodule symbiosis, and the nitrogen that is derived is called symbiotically fixed nitrogen. And this process is a very tightly regulated process that begins when the plant realizes that it doesn't have enough nitrogen in its environment. I'm sorry, there's a cat on my lap. Then it's when the plant realizes it doesn't have enough nitrogen, it starts secreting signaling factors such as flavonoids, which are perceived by rhizobia in the soil, which in turn secrete nod factors. And the first sign that these plants have perceived nod factors is the swelling of the root hair tips, as you can see in this image. And then these rhizobia attach to the root hairs, and with the bacteria here are stained in blue. And then the root hairs then start curling around them and trapping these bacteria in microcolonies. The plants then form these infection threads, which are extended plasma membrane, through which these bacteria, as they continuously divide, they move down these root hairs towards the growing nodules, which forms just below these successful infections. So in Medicago, the plant that I study, these nodules are indeterminate nodules because they possess a meristem, and these nodules are pink, elongated in their mature form, as in this image. There are, of course, determinate nodule forming plants as well, which lack a meristem, as is shown in this image. So we've made a lot of progress, and we've made a lot of great discoveries in legume genetics. And much of what we know is thanks to these two model plants, Medicago truncatula, which I study, and Lotus japonicus. But there are also a lot of genetic resources available for two of the crop legumes, which are soybean and common bean. So together, these four legume species form the basis of all the genetic discoveries that have been made in the last 20 years. So 2019 marked the 2 decades of legume genetics. And since the first mutant that was found in the symbiosis pathway, NIN, was found in 1999. So what we did in 2019 is we set off to try and catalog all the genes that had been found, which were required for functional symbiosis and for which we had proof via genetics. And what this image shows is the discoveries that were made over the last 20 years in the two model species, Medicago truncatula and Lotus japonicus. So I drew this as a timeline. And what it is exactly is what it shows are these groups of mutant populations that were developed from 1999 to 2019. And initially, in the late 90s and in the early 2000s, a lot of the mutant populations that were produced were derived from gamma ray mutagenesis, EMS mutagenesis, ion beam mutagenesis, and fast neutron bombardment. So this led to several successful forward genetic screens, and we identified several new factors which are required for this process. And in the late 2000s, then what we started doing was also developing populations such as the TILling and the tDNA insertion mutants, which were then used for reverse genetic screens. And then antisense RNA and RNA interference continued to be used across our community, but they typically have a lot of off targets. But arguably the most important populations for identification of important nodulation-related genes were two major populations. One is the TNT1 insertional mutagen population in Medicago, and the lower one insertional mutants in Lotus. And 2016 onwards, we've also started adopting CRISPR-Cas9 mediated gene editing to perform reverse genetic confirmation of target genes. So when I analyzed and cataloged all these genes, what I found that there are over 206 genes which have been shown to be important or required for formation of nodules and for successful nitrogen fixation. And we found that, interestingly, most of these genes required for symbiotic nitrogen fixation are also present in non-legumes. And several of these common symbiotic genes that we found were also required, were first evolved for mycorrhizal symbiosis. And since almost 80% of land plants can form arbuscular mycorrhizal symbiosis, it seems that these genes were recruited into the root nodule symbiosis process to interact with rhizobia. We also found that there were several differences within legumes, particularly the presence of nodule cysteine-rich peptides, which was restricted to galligoid legumes such as Medicago truncatula, but they were absent in determinate nodule-forming legumes such as soybean and lotus. And we realized that many genes that have acquired roles in nitrogen fixation is by expanding the gene family or by neofunctionalization and by gene duplications. So anytime there were these gene expansions and gene duplications, it was harder to find their phenotype because we had to then identify single mutants and double mutants. And so this resolving gene redundancy has been quite hard. So of all the processes that we cataloged and characterized, the one that I'm most interested in is long-distance signaling. And long-distance signaling in legumes is very well characterized. It involves two families of peptide hormones. One is called the C-terminally encoded peptides, or the CEP peptides, which were shown to be positive regulators, or they are the N hunger signals. And so these are produced by roots when they don't have enough nitrogen. And these are then transported to the shoot, where they are perceived by a receptor such as the CRA2 receptor, which then triggers a movement of CEP downstream peptides, mediating and upregulating expression of nitrogen uptake transporters. Because this nitrogen uptake process is very energy consuming, and nodule formation itself is energy consuming, there are also negative regulators called CLE peptides, which are clavatid-3 endosperm related peptides, which are produced when rhizobia are present. And these are also root derived signals, which are then transported to the shoot. They are perceived by a complex of receptors, which include the SUN, clavatid-2, and the corina receptors, which then trigger the formation of shoot-derived signals, which inhibit the formation of further nodules. So while I have told you of these two very important peptide families, I haven't really introduced peptide hormones. So what are these peptide hormones? Peptide hormones are 5 to 65 amino acid long residues of amino acids, which act as hormones just like the classical hormones, auxin, cytokinin, etc. They are active at very low concentrations, and they can also serve as receptor ligands. But since they are products of the mRNA, we know that the pre-pro peptide has an N-terminal signal peptide, which directs it to the secretory pathway. The middle variable region we do not have a function for yet, and the C-terminal region is where the signaling peptide is encoded, and it may contain several different post-translational modifications. So since these peptides are encoded by genes, it's very easy to study which ones are important for a process, and that is simply by looking at their gene expression. So that's what I started with. I looked at the RNA-seq data that was available, and we looked at the expression of more than 1,800 peptides that are encoded in Medicago and compared their expression to uninfected roots with nodule bumps at 4 days post-inoculation, 10 days post-inoculation, 14 and 28. And what I did then is take the average expression of all the different 1,800 or 2,000 peptide coding genes at all these different time points and divided them by the average expression of the gene in uninfected roots, and those that were very highly induced during nodulation or even were specific were what I then studied further. One of the most highly induced peptide family was the PAMP-induced secreted peptide family. And this family consists of 13 members in Medicago, so PIP1 to PIP13. And 5 of them were induced strongly at 4 days post-inoculation in nodule bumps, and 4 of them continued to be maintained in the nodule at all stages of nodulation. So when I cloned their promoters, I could see that PIP1, 7, and 13 were also induced in root hairs, which were infected. So here in magenta at the rhizobia stained, and blue is the GUS stain for the promoter. But not only in root hairs, when I looked at later stages, I found that all 4 of the PIPs, PIP1, 5, 7, and 13, were also expressed in the zone 2 of the nodule. And this is not surprising because in 2015, we conducted this extensive transcriptomic studies, and we found that all the genes that are expressed in infected root hairs are also present in the zone 2 or the infection zone of the nodule. And these genes, which are common to both these tissues, comprise a core set of genes which underlie infection processes during root nodule symbiosis, indicating that these PIPs are a part of these core set of genes, which had been overlooked by microarray studies. Come on. Okay. So what about peptide function? What were they doing during symbiosis? So normally what you would have to do is to study genes. You make mutants, you get mutants, you make overexpression lines, and that can almost take a year or more. But since these were encoding peptides, we had another tool to work with, which was to synthesize peptides. And there are some advantages and disadvantages of using synthetic peptides. The advantage being it's a very quick method for screening, so I could find results within 2 weeks. It can be applied as a solution very easily. It's relatively cheap, and it's non-GMO. But the disadvantage is still that its effectiveness varies between experiments, so therefore it's not a replacement for genetic confirmation. So what I did, I started by making these peptides and synthesizing these peptides, and I also was able to tag them with a fluorescent probe. And what you're seeing here is a nodule section of a 14-day-old nodule, which I dipped in fluorescent tagged PIP7, and then I cut it open and looked at it under the confocal microscope. And so what you see here in red, these red dots are PIP7, and this is the bright field of the nodule. And
sorry to interrupt you, we have 2 minutes.
Thank you. Yeah. Sure. So, and what we could see was that these co-localized exactly wherever the infection was present. And so these red dots are peptides which have been internalized into the nodule cortex. So when I also added these to the media and tested them upon infection with rhizobia, and what I saw that if you added too much of the peptide, it reduced nodule number, but it had no effect on lateral root numbers, whereas this is whereas every other peptide which has been studied also affects lateral roots. And like I said, peptide application is not a substitute for in vivo studies. So when I overexpressed these genes, I was able to see the exact same phenotype, which is a decrease in nodule number. But what happens in the absence of PIPs, right? So to answer this question, what I did was I mutated all 4 of them using CRISPR-Cas9, and I was able to find a phenotype. And what we saw that just like the overexpression in the quadruple and the triple mutant lines, also there was a decrease in nodule number. So what this suggests is, and what I'm concluding, is that an optimal concentration of PIPs is required for nodule formation. If you have too little, you have reduced number. If you have too much, also it's not an optimal amount of nodules. So with that, I'm going to summarize what I showed you today. I showed you that the last 2 decades have seen amazing progress in the discovery of genes and mechanisms underlying symbiotic nitrogen fixation. And I also showed that genome editing is helping reveal the activities of multigenic families and short ORFs, such as the PIPs, whose involvement in nitrogen fixation have been obscured by genetic redundancy. And thank you all for listening. Most of this work was done in Professor Michael Udbari's lab at the Nobel Research Institute, which I continue on at Tennessee State. And I'd also like to advertise an MS position and a postdoctoral position for anybody who would like to work with me. Thank you.
Thank you, Sonali. Fantastic presentation. So we have a question for you because you told us that it's very early in the morning for you, 3 a.m., right? So we have asked, there is one question which we want to take quickly. This is from Anthony from Kenya. He writes, great presentation, Professor Sonali. Could you please explain how yellowing symptoms, fluorasis of plant viral diseases affect biological nitrogen fixation, as shown in your slide on maize crop?
Sorry, I didn't get that question.
The question is how the yellowing symptoms caused because of viral diseases would affect the biological nitrogen fixation.
Oh, okay. So what are the interactions between? Yep. What are the interactions between viral infection and? So I would imagine that it is detrimental because the plant, if it's trying to fend off defense responses, and there's quite a lot of study on roots. If you look at disease, when plants have been infected with pathogens and symbiosis. Okay. But I would imagine if the plant does mount responses, defense responses, it would tend to block symbiosis, and it would be detrimental. But I guess there's a lot of literature that it does not come into my mind right now.
Fine. Thank you, Sonali. Thanks. Thank you. Also from the IAB, right? So it was a nice presentation, and there'll be more questions, I think so, as we go along. Now, moving on to the presentations by 3 young professionals. To start with, first we have Asina Beyter from Izmir Institute of Technology, Turkey. Her presentation is on characterization and association mapping of morphological traits in Faba bean. So we have the video, am I correct? So can you start playing it? Roop Kiran, Navneeta, can you start playing the first video by Asina Beyter? Yeah, thank you.
Hello, everybody. First of all, I want to thank you for this opportunity. It's nice to be part of this event. I will present to you our study, which is about characterization and association mapping of morphological traits in Faba bean. As we know that Faba bean is an important crop due to its high yield potential, nutritional dense grains, which are rich in protein, carbohydrate, and fiber. It's also low in lipids, and it is good provider of vitamin and energy. Therefore, Faba bean is used as a cheap and healthy source of protein, especially in many developing countries where the people cannot afford to buy the meat easily. Germplasm collections are important source of diversity. They can be used to enhance the yield, quality, and other morphological traits. When those germplasms are evaluated for agronomic traits, stress-related traits, quality and yield traits, then those information can be used for breeders to select the most suitable genotypes and parents and the cultivars. So therefore, in this study, first we aim to characterize 26 morphological traits in 114 different following genotypes, composed of 61 landraces and 53 cultivars. They were collected from different locations in the world. And then we also wanted to study the correlations between those traits. And in the end, we want to perform the association mapping studies for those traits using 100 single-sequence repeat markers. Our germplasm was evaluated for, as I said, 26 morphological traits containing vegetative traits, inflorescence traits, seeds, and pod traits. And our germplasm was evaluated for 2 growing seasons in Gülbahçe in Izmir, the west coast of Turkey. And then we performed the genotypic analysis to identify the polymorphic loci within the germplasm using 100 SSR markers. We also used the fragmental capillary electrophoresis system, which has quite high resolution, up to 3 watts per differences. And in the end, we obtained 711 polymorphic loci within the germplasm. After that, we performed association analysis using Tassel software between polymorphic allelic data and the phenotypic data. We tested general lineal models and the mixed linear models corrected with population structure and Kitch matrix, but based on the results, we found that the best one was general lineal model for our genotypic data, and we continue with these results. First, I want to mention briefly our characterization results. When we look at our data, we saw that some traits show really high heritability. These are number of pods per plant, seed weight, and seed yield. And also some of them showed the high diverse within the population, which are number of pods per plant and seed yield. We also obtained significant positive correlations between many traits. For example, we observed significant positive correlation between pod length and seed weight, pod angle, seed color, pod shape. And we also observed a significant positive correlation between the pod number per plant, which is one of the highest growth sensitivity in this analysis, and seed yield. This result is expected because pods per plant is a major yield factor in Faba. Also, we observed significant positive correlations between stipple spot pigmentation and pod with petal color and seed hilum color, which indicates that plants with stipple spot pigmentation always had spotted twin petals and usually had black hilum. We also observed that more horizontal and pendant pods tend to be longer and have larger seeds. Interestingly, we observed significant negative correlations between seed shape and 100 seed weight, pod length, pod angle, and seed cut color. So based on our results, we conclude that rounder seeds are often smaller than flat seeds, and flat seeds were more likely to be lighter in color and found in shorter and pendant pods. In our germplasm, we observed that there was high diversity for seed pod color, from black to white, and also we observed a significant positive correlation between seed pod color and pod stipple spot pigmentation and mid petal color, which indicates that plants with colored stipules and petals tend to have darker colored seeds. As a result of our association mapping studies, we detect 75 significant marker trait associations for 20 of 26 characters, of them 18 for vegetative traits, 11 for inflorescent traits, 13 for pod traits, and 33 for seed traits. Their PV value will range from 10 to 22%, which means they were major effect QTLs, and they would be useful for marker selection programs. And also we observed a marker colocalization for some traits, which indicates possible linked genes or pleiotropic effects. For example, those 5 marker loci were associated with both full spot pigmentation and wing petal color. 2 of them also were associated with hilum color, and 1 of them was also associated with both hilum color and seed coat color. So we conclude that because all these traits are related to organ color, this is likely that these are examples of pleiotropy. As a conclusion, in general, we conclude that phenotypic mass selection has been often inefficient when applied to fava bean using only yield traits. And also it has been reported that single selection of yield components has resulted in low yield stability in fava bean breeding programs. So instead of these combination of morphological characters that are indirectly related to yield components should be integrated into fava bean breeding programs. And also besides the regular breeding strategies, molecular marker approaches should be integrated to the breeding programs to maximize the success of yield improvement. As a result, we can say that our study is one of the largest association mapping studies based on large number of morphological traits and diverse fava bean genotypes. So based on our results, all significant QTLs were major effects on the traits which would be useful for marker selection on those traits. And of the identified significant loci, those for seed rate and number of the stems could be the target of marker selection for increased yield. Thank you for your attention. I would want to thank to my supervisor, Professor Dr. Frey and Professor Dr. Dolnar, and to my colleagues. And if you're interested in our studies, if you want to contact with us, please do not hesitate to write me. You can see my email addresses in here. So again, thank you very much.
Thank you, Asina Baiter, for that wonderful presentation. Next, we move on to another presentation, again on legume nodulation for biological nitrogen fixation by Chickpea Mung from University of Florida, USA.
वीडियो का साउंड है
Yeah, we're checking.
No, screen sharing is off.
Yeah, hello. Hello.
डबल क्लिक कीजिए स्क्रीन वन मिनट आप पोस्ट क्लेम कर लो पोस्ट क्लेम कर
I appreciate this opportunity to share the research in our lab here on the allele-specific expression of a transcription factor gene controlling peanut maturation. The non-modulating peanut plants were firstly discovered at the University of Florida here by a peanut breeder, Dr. Gorbet. He made a cross between these two lines, which produce nodules normally. And until F3, he started to notice a few peanut plants do not produce nodules. So by the F7 generation, he selected 2 pairs of sister inbred lines, each containing a NOD plus and a NOD minus plant. We applied a forward genetics approach to identify the genes controlling peanut nodulation, and through the map-based cloning and the complementation test, we identified 2 genes controlling the peanut nodulation in this background. And both genes are NSP2 genes. They're grass family transcription factor genes. If you don't know yet, peanut is an allotetraploid with 2 subgenomes, A and B subgenomes. So these 2 genes were located on the A and the B subgenomes respectively. So they're actually homologous. So the gene located on the A subgenome, we call the A gene, and the other one called the B gene. In this case, the A, big A, wild type allele, it is produce a functional transcription factor protein with 513 amino acids. The mutant recessive allele, small a, had nonsense mutation at the glutamine codon there, and resulting 224 amino acid peptides, which needs most of the functional domain. And the big B wild type allele can code a protein of 516 amino acid transcription factor, which is a functional protein. And the recessive small b allele, a mutated one, has had a deletion in the reading frame and called the early stop codon, resulting a peptide of 113 amino acid meets almost all of the functional domains. So with that, now we know the genotypes of all of the individuals in this pedigree, and the PI line has a homozygote dominant B gene genotype, and the UF line has a dominant homozygote A genotype. So from here, we can see as long as the plant has only one dominant gene, it will produce nodules normally. And the F1 plant is a heterozygote at both loci and produce nodules normally. And the F2 is segregating into 9 different genotypes. And all of the genotypes here, the right 7 genotypes in red color here, they all produce nodules, as we confirmed this already. And the recessive at both loci, homozygote recessive individual, do not produce the nodules. And this one, the highlighted genotype here with the heterozygote at the B locus, very interestingly, some of the plants do not produce nodules, and some of the plants do produce nodules. So with that, we further investigate the inheritance of this gene. And if a heterozygote plant at this locus self-pollinates, produce the population with 3 different genotypes, homozygote, dominant, heterozygote, and recessive. homozygotes in a 1:2:1 ratio. This has no problem. However, the phenotype here, this one produces nodules, and this one, some of the plants do produce nodules, and some of the plants do not produce nodules. And this one we know do not produce nodules. We confirmed this. So in that case, the phenotypic segregation ratio never be a 3:1 ratio. Instead, it is nearly a 1:1 ratio. So this, no matter what this phenotype is not plus or not minus, is always segregating in a 1:1 ratio. And we confirmed this. Here is actually an example of the field observation in 3 different years of the heterozygote self-pollinating population. It's always nearly a 1:1 ratio in terms of the phenotype, and never follow a 3:1 segregation ratio. However, the genotype segregation ratio is always a 1:2:1 ratio, which means the allelic separation or segregation is not distorted. So that is only the determination of this the phenotype. The heterozygote plants, some of the heterozygote plants can be not minus, do not produce nodules, even though the dominant allele is there. So the dominant allele in the not minus plants could be either like not expressing or silenced. And so we detected the big B and the small b allele expression in the heterozygote plants in both not plus and the not minus heterozygote plants. And we designed the allele specific caps markers and got the cDNA from the rhizobia infected roots and did the RT-PCR. So the results show that in the not plus heterozygote plants, instead of both allele expressing, only the big B allele expressed. And in the not minus heterozygote plants, it's only one small b allele expressed instead of both allele. And we got the cDNA from more roots at different stages for RT-PCR, and the story was the same. We also validated with the Sanger sequencing of those genotypes. So the conclusion here is the big B allele was only exclusively expressed in the not plus heterozygote plants, and the small b allele was solely expressed in the not minus heterozygote plants. We retrieved the phenotypic results of the previous published reciprocal crosses of the F1. We found that when the big B genotype as a female parent and the phenotype of the F1 were mostly dysfunctioning for nodulation. However, when the big B genotype as a male parent, all of the F1s produce nodules normally. So collectively, we can see that if the big B allele is contributed by the female parent, it is not expressing in the zygote. And this allelic expression of the B gene could be due to the maternal imprinting. So it's depending on the origin of the allele. Only one allele derived from the male parent is expressing in the peanut plant, and the other allele from the female parent is balanced by imprinting. So to test this, we collected tons of flowers from peanut plants of different genotypes and phenotypes to dissect the ovaries and the pollen for qRT-PCR. And the results showed that the gene expression level in the peanut pollen was significantly higher than that in the ovary, no matter what the genotype, homozygote or heterozygote or phenotype, not plus or not minus. It's the same thing. So we can conclude that the B gene is imprinted in the female gametes. So the take-home message is the NSP2 gene at the B subgenome is maternally imprinted. And to our knowledge, this is the first genome imprinted gene identified in the peanut genome, which may be under the epigenetic control. And with that, I appreciate all of the lab members and the collaborators contributed to this research and the funding agents. And thank you very much for your attention.
Thank you, Jingping Wang. Very interesting to learn that maternal imprinting. Yeah. So we will move on to the next presentation for the day. Sokia Konde from CIRAS. It's a regional research center for agriculture, which works on plant adaptation and drought. She would be talking to us on the development and characterization of a unique genetic resource for alum mining and peanut improvement in Africa. Over to you, Konde.
Hello, I'm Sokia Konde. I'm PhD student at the Regional Study Center for Crop Improvement and Adaptation in Senegal. I am co-supervised by Dr. Jean-François Ramy and Dr. Daniel Fonseca. My research activity focuses on the study and valorization of genetic diversity within the peanut crop compartment. I'm going to present to you a first chapter of this activity called Overview of Grown-up Genetic Diversity held by 10 breeding programs on 9 countries in East and West Africa. First, I am going to introduce the reasons that led us to take up this research question. I will then develop the method used before presenting the result, the conclusion, and the perspective. Within the cultivated peanut, we have an important agromorphological diversity. Gen bank and breeders hold a part of this diversity. At the same time, there is a low allelic diversity caused by tetraploidization and domestication events. So we have narrow in genetic base in peanut cultivated compartment, and it is a serious problem in agriculture. To improve this genetic base, we have to look for the maximum of resources to build a large panel in order to find allele of agronomic interest. For this, we have drawn on the diversity already mobilized by the selection program. The plant material is an assembly of peanut germplasm. Indeed, each research institute visible on the map has provided about 10 seeds for each accession of the best of its breeding program. So, from June to October 2018, we received at CIRAS 1,132 peanut accessions. These genotypes were grown in the greenhouse of CIRAS. Only one plant is grown for each accession, and 1,050 have germinated. The harvest seeds are multiplied, and one part is stored in the physical gene bank of CIRAS, and the other part is sent to Uganda. The DNA of 1,049 accessions is extracted from young leaves, then purified and sent to the Georgia University for genotyping. This collection was genotyped with 47,837 SNP from the Axiom Arachis 2 ships. Analysis of the genotyping data identified 8,911 good quality polymorphic SNP that were used to describe the genetic diversity and structure of the use of the accession. Using expected heterozygous, DAPC analysis, ACP analysis, and admixture. We also defined a core collection based on breeders' knowledge of the germplasm and the result of the genetic diversity analysis. This core collection is being evaluated in various environments in West, East, and Southern Africa for traits related to disease, yield component, and nutritional quality. Now we will present some result of this study. Here we have the result of the DEPC analysis performed on 6,205 SNP and 149 Arachis individual. In A, we have a scatter plot of this principal component, of this discriminant principal component. Spot represent individual, color indicate genetic cluster. We observe the presence of 4 groups within the collection, and we distinguish 2 large groups separated by the ordinate axis corresponding to the 2 species of Arachis hypogea, namely Fastigiata and Hypogea. The x-axis separates the subspecies Fastigiata in three groups. The first one constituted exclusively of Spanish in red. The second one constituted of a mixture of Valencia and Spanish, and the third one constituted of an interspecific individual resulting from a cross between a wild and a cultivated. The structure observed in this panel is mainly explained by genetic cluster, which have some correspondence with botanical variety and marker type. This table compares the result of the previous DAPC with a DEPC where the number of clusters is 10. It shows the genetic substructuring that subdivides the commercial type. Here we have the result of the PCA analysis performed on 6,205 SNP and 1,049 RHS individual. On the axis, we have the principal component 1. On the ordinate, we have the principal component 2. When we colored ACP with the country, at the first side, there is no geographical structuring. But with this graph in sector, which concern only the Spanish 2, we see that there is a geographical substructuring. In fact, 98% of this group is made up of West Africa, 74% of whom are from Mali. The botanical type exploited vary from the one country to another. For example, in Niger, the most exploited botanical type is the Spanish one. We have very few Spanish 2, 3, 4, Valencia, and almost no Virginia. Here we have the observed heterozygosy, or expected heterozygosy, and inbreeding coefficient were calculated using data R package. Expected heterozygosy is a common statistic for accessing genetic variation in population. High heterozygosy means lot of genetic variability, and low heterozygosy means little genetic variability. Thus, the country is where we have the less diversity are Mali and Niger, so we'll gain in diversity by receive the core collection. The country where we have the more diversity are Senegal, Uganda, Zambia, and Malawi. To conclude, the structure observed in this panel is mainly explained by genetic. In effect, we have genetic structuring and geographical substructuring. Thank you for your attention.
Thank you, Sokia Konde, for that presentation. Now, next is our invited speaker, Professor Kadambat Siddique from University of Western Australia, who will be speaking to us on adaptation of chickpea to terminal drought, physiological and molecular approaches. I mean, most of you in this group know Professor Siddique, but then it's my honor to introduce to you all today. So Professor Siddique has over 30 years of experience in agricultural research, teaching and management, both in Australia and overseas. And during his tenure, he has developed national and international reputation in agricultural science, especially in the fields of crop physiology, production agronomy, farming systems, genetic resources, breeding research in cereals, pasture legumes and grain legumes, and oilseed crops. And he's a prolific writer. He has published more than 775 scientific papers and book chapters, and he's been the highly cited agricultural researcher in the last 4 years, 18, 19, 20, and 21. Professor Siddique was designated by United National FAO as Special Ambassador for the International Year of Pulses to 2016. And he is a fellow of several professional academies and also recipient of national and international awards. And Professor Siddique has also trained several honors, MSc and PhD research scholars, and developed extensive network of scientists within Australia, and has established diverse range even overseas that include China, India, Turkey, Syria, Iran, Iraq, Saudi Arabia, Oman, Malaysia, East Timor, Nepal, Bangladesh, Pakistan, Europe, Canada, and USA. So it's a pleasure to have you here, Professor Siddique. Over to you.
Thank you, Janila. Can you hear me? Can you see me?
Yes.
Yes, and my slides are good?
Yes, they are good. Please go.
Oh, good. So once again, I thank you and Judith and the organizers for inviting me. I know the time is short. I'll do the justice. So I'm going to talk about adaptation of chickpea, which is an important crop for you in ICRISAT, and for India. We heard from your DG. The terminal drought, again, a number of speakers mentioned about, in fact, the DG has mentioned about the climate change. The COP27 meeting is going on. We are really reaching a critical stage. So what are the physiological, molecular, and breeding approaches? I'll use some case studies, including the work done in ICRISAT and Australia. So the second slide, if you can see that. Janila, can you see that?
Yes, we can see.
Okay, so the background, then I will just talk about the dryland agriculture and how climate is affecting Australia. Australia is the driest human inhabited continent in the world. And then some field work we have done and then taken into the controlled environment, then the breeding strategies, and then more recently the work we have been doing with ICRISAT, particularly with Rajiv Vashni and also with the Bharadwaj and others at a team of people from IRI and other places. So just want to acknowledge the funding from various sources, Australian sources, the university, the departments, then AISRF and so on. And Janila just mentioned, I had the privilege to work with 14, 15 countries, the International Year of the Pulses, and clearly the pulse production has gone significantly high in many countries, particularly in India, thanks to ICR and ICRISAT and other organizations. I won't go into the details, but I would like to draw to your attention this paper some time ago we published. The main point I want to emphasize is global investment in and pulse research development and extension is too low compared with the cereal crops, the major cereal crops. So we receiving only about 175 million per annum, 13 or 15 pulse crops. So we really need injection of funds to this crop in variety of topics because we need the pulses in the system. Again, there has been a lot of emphasis by Sean and your DG about the importance of pulses, particularly in relation to the sustainability. Again, I won't go into the details. You all know self-pollinated, originated in the Fertile Crescent, grown in 50 countries, mainly in South Asia, and it is really second among the top food legumes in the world. Two types, Desi and Kabuli, high protein, very good quality, human consumption. You don't need much to do with that. And the most important in this context, significant amount of nitrogen is fixed, and those nitrogen is left for the succeeding crop. But also, crops like chickpea have the ability to excrete organic acids through the root and then solubilize the soil-bound phosphate, iron, magnesium, and so on. So there's a lot of benefits. So about 13 million tons produced and about 9.6 million in India. So India is a big leader and others are also there. Coming to Australian situation, it's a new crop relatively, and my PhD was the first PhD on chickpea in Australia, so I can take a little bit of credit there. But we continue to rely on importation of germplasm from ICADA and ICRISAT, India, and so on. And we had an industry going reasonably well by late 90s, and the Ascochyta blight came and completely wiped out. Now we have a good breeding program. We have our own varieties, and it is one of the largest, and we produce about 1 million, 1.2 million tons of chickpea, and majority were exported. And this is the grain belt of Australia. Virtually, we can grow chickpea in all those golden or yellow triangle. This is about 300 to 600 millimeter rainfall isohytes. So it's entirely dependent upon rain-fed agriculture, except up in the north, in a place called Kananara, we have an irrigated agriculture, which is large garbanzo, a small industries operating there. Coming to Western Australia, if you look at the map, you can see it's a cold, wet winters and dry summer. So we have a Mediterranean type. As you move to the east, it's more evenly distributed. As you come into the north, Queensland and New South Wales, it's more like the northern Indian conditions. And our grain belt is a low rainfall, 250, 325, 325 to 450. So very low rainfall environment. Hence, drought and heat adaptation is absolutely essential. Very quickly, the climate is changing in Australia. And if you look at the 1990 as the base and look at the 2030 figure, you can see the temperature is increasing and rainfall is declining, except up in the north, and then evaporation is increasing. So no doubt that Australia is facing climate change. And this is very clear, the southwest of Western Australia, where our agriculture is located. You can see from 1900, continued decline in rainfall, and we are seeing very, very dry years in recent years. It's not only the rainfall. If you look, 0 is the baseline, and we had many more colder days during the 1910 up to 1975. Then we are beginning to see hot events, which is really affecting the production. So in my talk, I'm going to focus on terminal drought, the soil moisture stress that occurs during flowering, pod filling, and seed development. So that's quite similar in India and many other parts of the world. So a major constraint, more than 80% of the global chickpea area is affected by one form or other form of terminal drought. In many cases, heat stress is also affecting. So what we've done is that we have been doing and breeding, selecting, collaborating, etc. So our main objective was to identify chickpea genotype resistance to drought in a Mediterranean type environment, meaning dry summers and wet winters leading to terminal drought. And we wanted to underline the physiological mechanism and work with the breeding programs. So we used two approaches. One is started evaluating number of promising lines in the field, and then taken selected lines from there to understand, unravel the mechanism involved. So the map of is here is Perth, and locations like Bindi, Kundradin, it doesn't matter. 2012, water limited. 2013, again water limited. A different number of lines from 2000, we distilled into 180, 62, and 2014, 15, we then taken into the glasshouse experiments. So the yield summary from the large field evaluations of place called York, and as you see, there is significant difference. The main point is that some lines showed consistently high yield. So for example, the red I marked here, and the blue line is showing low yield. Of course, the ranking changed, but generally speaking, we were able to get a good handle on the field data. So from there, we selected 10 or 12 low and high yielding lines, and then we moved into large bin system. This is the bin system, like a rubbish bin, which can hold up to 100 to 110 kilograms of soil, as opposed to people that work on small pots. I am very critical about people working on small pots on drought, etc., because it doesn't really mimic or at least simulate the field conditions. So then we had two treatments, well watered, about 80% of field capacity, and water stressed around the early podding time. And we have tagged the fecundity, the fate of those flowers during drought very systematically. You can see the thousands and thousands of tagging and the dates where the flowers formed, where the pods formed, etc. And we also developed a balance so that we can weigh the transpiration loss. So it's basically an hydraulic lifted. Any small person can use one hand and then weigh with an accuracy of about 10 grams. So this has really helped us to understand and study the transpiration loss. Again, I know with the time, I won't take a lot of time, I'll go. So here is the soil water content, percentage of the field capacity, days after drought treatment. You can see the drought started at early podding time, and then well watered is maintained. different varieties, different breeding lines. You can see Neelam is one of the variety I have released with one of my colleagues here, and these are some of the breeding lines. So basically what we are seeing is that the pattern of water use varied. For example, Neelam used water more conservatively in order to produce use that for better seed, for pod and seed filling, whereas others were more opportunistic. They continue to use water and has got implications later. So you can see, for example, the genotypic difference 24 days after stopping water, nearly 1 month. So if you do that in a small pot, it will not last more than 10 days. So this is the beauty about the larger volume of soil, more or less like field situation. Neelam is our variety. Sika is one of the most susceptible line. So well watered on the left, and then you can see on the right. You can see the soil water content was similar among different genotypes at 2.6% weight by weight. That is equivalent to 13. And there is still, in the case of Neelam, continued pod production happening under drought. That's the picture. We went and looked at the number of reproductive organs. So you have got the flowers, total pods, seeds, open circle, open shows the well-watered, and the closed one shows the water stressed in different genotypes. Basically, genotypes behave differently. We were able to follow the fate of the flowers and the pods produced during that 24-day period of the drought. We know that the flower, the pollen tubes in the style when soil water was reduced to 0.25%, and there were some pollen tubes growing, but it was affected. So we went and looked at the pollen viability and pollen germination, and the pollen viability in well-watered conditions stayed, and whereas in the case of droughted conditions, it declined drastically. And there were genotypic differences for that. And the pollen germination was also shown there. So our analysis shows that the pollen tube reached the ovary in all flowers when soil water content was reduced from 80 to 25% field capacity. So we conclude that increased flower abortion, pollen tube growth into ovary could not guarantee the success of fertilization. Possible inhibition on ovary fertility may be a case. I don't think you can see this pollen tube is growing here. So it could be short of carbohydrate supply, the hormone may be the hypothesis we have decided, we have coined. So here I'm showing the fraction of transpirable soil water, the number of reproductive organs, flowers, pods, fill pods, and seeds. So basically we were able to show that flowering and total pod stopped at 0.2 fraction of the transpirable soil water. Whereas the seeds were protected better, like any young babies, we protect them. So they were ceased at 0.6 there. So they in fact stopped at a higher there. When the stomatal conductance and transpiration began to decline. So this suggests that pod and seed abortion was induced by reduced assimilate supply. Then we looked at a range of abscisic acid and by tagging, and this was done using the flowers. And here you can see 9 days after flowering this pod, and also see, for example, the 16 days after flowering, the water stress test treatment, almost all the pods dying, whereas the well-watered conditions we can see. From there, we looked at the pods developed from flowers tagged on the same day at 80% fill capacity, and look at the ABA content. ABA content, as soon as you water stress, WW stands for well-watered, increased here. And as we continue the stress to 50% field capacity, again has increased. And there were genotypic differences in response to ABA production in water stress. And then we looked at the ABA in the seed as well. So we have got pod wall, seed, days after flowering, 16, 23, 16, 23. Again, two sets of genotypes I'm shown here, well-watered and water stress. Again, you can see in the pod wall, ABA content increased, and at the 16 and 23 days, and the seed also significant increase in ABA content at water stress conditions. Of course, the drought reduced the seed size. This is a mean seed weight. The red one is well-watered, and the blue one is water stress in all genotypes. but the magnitude varied. If you look at the yield under well-watered conditions, you can see there were differences with this CICA line giving slightly higher yield and also this decline. But when we do the water stress, the whole thing changes. The Neelam gave high yield, whereas the CICA 912 just collapsed. So of course the scales are different here. This is up to 8 and the other one is 40. So there are differences coming in. We also looked at the relationship between seed yield and the pattern of water use. So if you look at the graph A, this is the seed yield of water stressed plants, and you can see that water use at different times. There are two linear figures here. One, the red one is 104 days after sowing, and the other one is 106 days after sowing. So what it shows is that if more water is used during early time, it is negatively related to the yield. Whereas if you look at the bottom graph, you can see that the more water used in the post-flowering days, this is 112 and 117 days, there's a good relation. So the pattern of water use drives the yield. We also done a significant amount of work on the root architecture. I won't go into the details. We have a platform here. We can quickly screen genotypes for. So for example, look at the differences in two genotypes, ICCV 95311 and another one, ILLC. There's a different type of root architecture. So the mini core or the reference set, we looked at it. You can see the taproot length is significantly varied from genotype here to here. And of course, we also looked at the smaller set of Kabuli in the reference set, which is about also significant difference. So basically, there is root rates are significantly different. So in summarizing this part of the story, drought affects the pollen sterility, possible fertilization failure due to ovary viability, or other factors, example, AB and sugar. And the flower abortion occurs, pod abortion, and cessation of the seed growth and leading to yield reduction. But the question is that does ABA and carbohydrate play a role? And that still needs to be worked out, and I believe there is quite a lot of work to be done in that area. Quickly moving on, Janila, I think many of this physiology has to be embedded in the overall picture. So this is one of the papers published by Rajiv et al., 2 papers. Genetic resources, we want improved germplasm leading to improved varieties. A lot of technology, the technology is available. So I would like the young people to focus not on the technology, but on the outcome, but use the technology, whether genotypes, the genes, the trait QTL, or the phenotyping. All is required. We need all that, and then we can use different tools available now and the emerging tools. So again, the collaborative work with ICRISAT, we were able to sequence that 429 chickpea accessions. Some of those data we have used in further studies. This is one of the study where we looked at Australian breeding lines on the top. Then we also looked at the Indian derived breeding lines, clear separations of the cluster there. And when we looked at some of these lines in the field, seed yield, and done some preliminary GWAS work, again, this is published, you can see that the seed grain yield here on the top, seed number, then empty pods, empty pod ratio, and we have also got early vigor score. So there are some interesting emerging regions containing SNPs significantly associated with the different traits are highlighted in this here. So that's promising. Now we also have a still ongoing project with India, ICRISAT, JNU, IRI, and others, and that project is looking at the functional genomics of chickpea to enhance drought tolerance. And again, of course, more recently, 3,000 or nearly 4,000 chickpea genetic variation mapped and cloned. So this will be very useful for further moving forward. Again, the most recent work by Ratwik and the PhD student with Rajiv and all of us and characterize the QTL hotspot, the Indo-Grecian lines where the physiological mechanism is shown. And of course, a number of varieties have been released. This is the partnership where the ICRISAT team partnered with the Indian National Program, particularly with the Bharadwaj and Co., and helped to release. Perhaps this is one of the finest examples in literature where physiology, breeding, genomics, genetics works together in order to produce improved varieties over the standard varieties. I won't go into the details because of the time. Again, the so-called super Anigeri 1. Anigeri is widely adapted, but it has got Fusarium wilt susceptibility, so new varieties have been released. So if you go back, the Ratwick's work, again, yield at different locations. What we have been able to show here is yield performance of chickpea endogress lines compared with the donor parent, which is the high root one, ICC 4958. So again, I won't go into the details. Gulbarga, Coimbatore, Vijayawada, and all those places, multi-location trials revealed the superiority of the endogress lines. And of course, now they are released as varieties, some of them. They went and looked at the leaflet size. Again, you can see the significant difference is denoted by the letters, and the donor one, ICC 4958, is shown here in relation to the leaf area. But the most interesting thing for me as a physiologist is that the transpiration efficiency. So here is the PUSA 372, BGM, and the ICC 4958, which is giving the root and other traits. And again, at different locations, you can see at different levels the improved transpiration efficiency through that endogress line. Then, of course, went and looked at the donor parents in red, recurrent parent alleles in blue, and then you have got the heterozygous regions, which is the validation of the diagnostic markers for drought adaptation in chickpea. And another spin-off effect was that the haplotype analysis of the gene across 1,548 Desi accessions showed some of the seed size. So it's just not the drought tolerance or the root work, but the seed size was also improved. Perhaps the seed size is also contributing to the high yield in drought environments. Janila, I would like to wrap up with all that. One of the summary paper, again, Rajiv led that with a number of us, shows that it's important that we need to look at a comprehensive approach. So maximizing genetic diversity, you can sequence, genotyping, phenotyping, simulation of crop models are important because these days we can even model gene-to-gene interaction, development of diagnostic markers, and then sequencing of the trait mapping and gene discovery. That will improve genetic enhancement. Large number of crosses needs to be made. This is where the interaction with the breeders has to take place. And we need to generate large segregating breeding population, early generation screening, genome-wide prediction, then of course, molecular assisted breeding program. But also, even in our lab, we can take up to 6 to 8 generations of chickpea and other legumes through rapid generation cycle, simply by good tissue culture, but also the use of LED lights to mimic the conditions. So then now genome editing is also available, but the traditional breeders need to look at the improved operational efficiency, appropriate design. All the biometrics have changed. The classical biometrics, nowadays we have many ways in which we can look at the data. The field book-based data recording, all automated, analytical and decision support tools. Use of breeding database. The platforms can share information between countries, individuals, and continents. Now we need to have the integration of the digital tools and technology across the breeding process. Otherwise, we will not be able to cope up with the situation. We want to rapidly get the good varieties to the farmers' hands, not 10 years since started the breeding program. But all that will not work if you don't have an efficient seed system and agronomic practices and the market access. So you may ask, why India just all on a sudden bumped up its pulse production, including chickpea production during the last 4, 5, 6, 7 years. That's one of the things was ICR and the government of India provided good quality seeds of the improved varieties at an accelerated rate through various programs. And of course, the support price and the market access is important. So we need to strengthen the formal and informal seed system, and we need to have a faster variety replacement. There was a time when varieties will never be able to replace. The committees after the committees, the bureaucracy, look, the farmers doesn't care about the committee. If the variety doesn't work, they will throw it out. So let us bring varieties with a better approach and let the farmers test it. Availability and affordable high quality seeds, this is a big problem because crop like chickpea or legumes need about 80 to 100 kilograms per hectare and the farmers cannot afford. And we need to look at the soil health, optimized agronomic practices, access to farmers, the market is very important. And that government of India done that. And of course that hurts countries like Australia, but eventually there will be room for all of us. So we need input from stakeholders, higher genetic gains and increased productivity. And that's a nice paper summarized in I think in Journal of Experimental Botany. Thank you once again for giving me the opportunity. I hope I haven't taken more time than allocated. Thank you.
Well, thank you, Professor Siddiq, for that excellent and scintillating presentation, as one of the participants has written in the chat box. Yes, I mean, thanks. Good morning. The DG Jackie Hughes has pointed out how the training in mathematics of Mendel helped him to work on the genetic ratios of biology. And today, when you're presenting, you touched upon how the different disciplinary teams should come together to translate the basic research into the applied products that would benefit the global community. Thank you for that. I will just have a quick question because I know that you need to move on to the next meeting.
I'll be here another half an hour. My meeting starts at 4 o'clock, so that's good. I'm good here.
Okay, but then let me take this question. So
please.
You talked about the drought tolerance breeding. Sanjuti from NIPGR India wants to know your thoughts on the roadmap to integrate nodulation in the chickpea breeding program. And I'm sure this is also important for drought conditions as well. Over to you.
Yeah, so I think it's a very good question. So we need to look at all aspects, of course, disease resistance, yield, etc. So what will happen in our situation is that the nodules are mostly in the top layer. In all cases, that is about 0 to, let's say, 20 centimeters. So earlier on, the moisture is not a limitation. Rainfall is coming, the plants are growing, they fix the nitrogen. That nitrogen is then available to the plant. So in a way, when you get very early drought, then the situation is bad. So then we need to look at the better nodulating type, better bacteria and genotype, etc. Sure, all research we need to look at the biological nitrogen fixation. If they don't fix, if they don't. In all our cases, we applied the rhizobia, whether it is the control plant or droughted plants, then we looked at. So we don't apply additional nitrogen. So simple answer is that, yes, there is an interaction between drought and nitrogen fixation. And you need to then define which condition you are looking at. If it's early drought, then it will definitely affect the nodulation and nitrogen fixation. And there must be genotypic variation both in plant, but also in rhizobia, strain variation.
Thank you, Professor Siddiq.
Thank you.
Yeah.
Thank you again. Thanks again.
Thanks for also staying just for another 30 minutes till we close the session. So now next speaker is again a young professional, Kritika Anbazhagan from ICRSAT India. She will be talking to us on capturing the genetic variation in Mung germplasm for crop vigor and canopy size using 3D cloud points. Over to you, Kritika. Can you play the video?
A very good morning to all of you. I am Kritika Anbazhagan, a researcher with the Crop Physiology and Modeling team at ICRSAT. It's a great opportunity for us to be presenting at the ICLGG conference today, and here I'll be talking about the work that we are doing with the World Vegetable Center on capturing the genetic variation in the mung bean germplasm for crop canopy traits. For this, we use the high-throughput phenotyping facility LICI-scan that was established at ICRISAT around the year of 2015. Within the presentation, I'll give a brief overview of the mung bean germplasm collection, primarily the genetic material, and the traits for climate adaptation, high-throughput phenotyping system itself, and the data analysis. The mung bean germplasm collection comprises of the many-core lines, a few advanced breeding lines. We know that the breeders have to invest several years We know that the breeders have to invest several years and resources in identifying varieties that would be released and adopted by the farmers. In the case for climate resilient crop types, they would have to manually characterize these phenotypes either visually or manually for crop adaptive traits. In this particular scenario, it would be necessary to have the phenotyping pipeline similar to what we have at ICRISAT. So here we have the sensor-based methods wherein we are developing protocols for using UAVs and LICI-scan for extracting component traits of adaptation, from where we can filter down a few hundred lines that can be used for detailed characterization for water use traits as well as agronomic traits across the crop cycle in different seasons, in different soil types, with different fertilizer application, etc. From here, we can handpick a few varieties with the desired phenotype for specific targeted environments for a much precise characterization of lines. Coming to the component traits of adaptation, what we mean by that are the root and shoot traits that contribute to biomass. So at ICRISAT, we focus on canopy traits, for which we have the LICI-scan and the LICI field system. So we look into leaf area index, leaf number, leaf growth, canopy vigor, all of which contributes to the radiation use efficiency, influences transpiration and transpiration efficiency, which result in the biomass that is accumulated by the plant, which influences grain yield further through grain number, grain size, or quality as well. The high-throughput phenotyping system at ICRISAT is called LICI-scan, and here we have 4,800 microplots that are stationary, and the scanners move around them in a span of 2 hours and capture these images. So we have noticed that within 4 to 6 weeks of plant growth, we are able to capture canopy traits that will enable us to make selections for further detailed characterization. For example, here we have pearl millet breeding populations, 3 sets of those compared against the elite lines. And we are able to see the digital biomass and the variation across these lines. The sensor-enabled high-throughput phenotyping system has been recently upgraded with the F600 scanners, which has this dual scan strategy for improving the resolution of the 3D point cloud images. And it also has RGB and NIRS sensors, which will enable us to look at not only traits, canopy traits related to climate adaptation, but also biotic stress and probably senescence and quality. We have field scales that help us look into monitoring water use and capture evapotranspiration and transpiration across different lines. And we have a weather station located there to look into the specific climatic conditions while the experiment is being. Once we have the 3D point cloud based digital plant constructed, several artificial intelligence based algorithms are applied to extract features such as leaf area, plant height, digital biomass, leaf angle, plant count, evapotranspiration, etc. We are now looking into leaf expansion, probably tiller number, and disease score as well. In summary, the sensor scans the crop, a digital plant is constructed, and several parameters are extracted from these images. With all the data sets that are generated from these 3D point cloud image extraction, we have big data sets to work with, for which we have a data analysis pipeline, wherein we look at pre-processing to ensure that we have good quality data for assessment. And then from here, we do a critical window selection, looking at heritability as well as variation. We do a huge and statistical analysis prior to clustering the phenotypes. Coming to the Mung bean dataset, we had 341 genotypes grown in the summer season of February and March in 2020. The plants were grown in 4 replicates and 4 plants per microplot were maintained. The graphs here indicate the plant growth across experiment. We are depicting the plant height here. So the left panel shows across the entire duration of the experiment, whereas the right panel indicates the window that we have used for data analysis. So this graph shows the gray lines are the mini-core lines, whereas the orange are the elites. And we can see the variation in 3D leaf area across these 341 lines. The 2 graphs here indicate the box plots. The left panel showing the elite and the advanced breeding lines, which ranges from around 70,000 to 12,000. Whereas the mini-core lines range from around 30,000 to 12,000 of 3D leaf area. And the plant height ranged from 130 mm to 310 mm across these collections. Similarly, we can see that the biomass that was harvested at the end of the experiment showed a large variation in the mini-core lines, ranging from 8 to 43 grams per plot, whereas that of elites were ranging from 17 to 34 grams. In summary, we had 341 lines screened in lazy scan for canopy traits in the summer season, and we see a large variation in these traits. We would now like to look at the response of these lines in different seasons, in Kharif and Rabi, and see how these lines respond differently. Prior to concluding, I would like to give a case study wherein the QTLs identified for plant vigor and water use traits and the chickpea lines co-localized with the same region identified by conducting experiments in 5 locations across 7 seasons using root traits, agronomic traits, and seed weight. Similarly, the QTL for water-saving traits in pearl millet, localized in LG2, was also identified with the LISI scan experiment. I would like to conclude saying that there's a huge potential for high throughput phenotyping systems in screening large germplasm collections rapidly and for selecting breeding lines for accelerating crop improvement programs. Thank you very much for your time. Have a good day.
Thank you, Kritika. Thanks for that nice presentation. We'll move to the next presenter in the interest of time. We have Nikki Sotsora from University of Nottingham, United Kingdom. She'll be talking on the QTL analysis of nutritional traits in winged bean. Can you start the video?
Hello everyone, my name is Nikki Sotsora. I'm a 3rd-year PhD student at the University of Nottingham, and I founded by Future Food Beacon to work on an underutilized crop called winged bean and explore its potential to be a new soybean for the tropics. As we're all aware of the Sustainable Development Goals of the United Nations, one of them is to end hunger and achieve food security by improving nutrition and promoting sustainable agriculture. And if we look at the statistics, 2/3 of the undernourished people across the world, they live in sub-Saharan Africa and Southern Asia, suffering by protein allergy, malnutrition, and macronutrient deficiencies that can affect their health by causing diseases such as marasmus and kwashiorkor. But this is expected if we look at our intake, our food intake. We heavily rely in a handful of crops, with maize, rice, and wheat providing 60% of our calorie intake. However, there are more than 20,000 plant species that they're edible. So we can focus on exploring other crops. There are crops that haven't received a lot of research called underutilized crops. These crops are of high nutrition value and they're indigenous crops, meaning that farmers and people are familiar with their cultivating practices. They also carry traits that make them resilient to climate change, as well as biotic stresses, which is quite important. Winged bean is one of those underutilized crops. It's a tropical legume that is cultivated mainly in Southern Asia. And as you can see, it has a protein content similar to soybean. Apart from the seeds that they have high protein content, also are edible, the tuberous roots, even the leaves and the flowers. As a legume, it's a good nitrogen fixator. It's all fertility and structure. My research is focusing on improving protein quality. About focusing on high protein content, we also need to improve the quality of the seeds, and this is determined by identifying the limiting amino acids, as well as lowering the level of the anti-nutritional factors. This way, we can improve the digestibility as well as the bioavailability of the protein in the seed. For that, I've used a population, an F2 population that has been generated from a cross between two winged bean lines, M3 and FP15. As you can see on the right, they vary significantly in the protein content, but also on the morphological traits, with FP15 pods being purple and also very significantly in the color, with FP15 having purple pod and seeds compared to M3. So it is important to identify the genetic variability in this cross and associate the genetic studies with the compositional studies to identify QTLs for winged bean genome. So what is a QTL analysis? It's basically a statistical method that links the genotypic with the phenotypic data. So for that, I've used 160 individuals to identify protein QTLs. As you can see, more of the individuals in the frequency protein graph, they would fall between the parental lines, M3 and FP15. However, there were still some extremes, showing that there's a transgressive segregation for the protein trait. And as you can see on the right, on chromosome 3, there's a significant QTL. And in that specific region the print and is under review to be published in a journal. I've also mentioned that it's important to identify the limiting amino acids. As you can see in the lipic barrel, by increasing the amount of the limiting amino acid, we can increase protein synthesis. And as we know, legumes have the sulfur-containing amino acids as limiting, like methionine and cysteine. You can see on the left, look at the essential amino acid composition. I've done the amino acid analysis using HPLC, comparing high and low protein winged bean lines to soybean lines. You can see that the percentage of amino acids resembles the one of winged bean, similar to soybean. But if we look at the amino acids as gram per 100 gram of dry seed weight, we can see that the amount of protein significantly affects the amount of amino acids, with the low protein winged bean line having less than half of the high protein winged bean line. You can also see that lysine, for example, in the high protein winged bean line, it's even higher in terms of composition compared to soybean. So it is important to take that into consideration when we're supplying winged seeds for animal feed. It's also interesting to look at the anti-nutritional factors, because it's part of the defense mechanism for the plants against fungi, insects, and herbivores. But these compounds can inhibit the absorption of nutrients, and they can even cause deleterious effects if the pulses are eaten raw. just to mention that there are two groups of anti-nutritional factors with protein compounds and non-protein compounds. It's important to note that protein compounds can be reduced by applying heat. And recently for winged bean, there has been some research on the transcriptome era, focusing on trypsin inhibitors as well as high and low condensed tannins. This work is really important because it can help identify genes that they can be used for improving based on breeding for lower level of the nutritional factors. But it's also interesting to look at a thinner seed coat, because having a thinner seed coat would increase the absorption of water and reduce the hard to cook phenomenon that is quite common among legume seeds. There are other methods, processing methods, to reduce the amount of anti-nutritional factors, with germination, removal of the seed coat, and soaking in water being the popular ones. It's also common to heat the seeds by autoclaving, boiling, or even microwaving them. As I mentioned, there are anti-nutritional factors that they can be deactivated by the application of heat. So that raises a question: if processing methods can reduce the activity of protein inhibitors, protein anti-nutritional factors, then do we also need to breed for a lower amount of protein inhibitors, considering that if we deactivate them, there are still proteins that can be used in the digestive tract. So it is important to have more research on underutilized crop. Looking at winged bean, there's still a lot of research that needs to be done to improve its quality and identify genes and molecular markers for further breeding selection. We are facing a lot of challenges at the moment with population growth and climate change, and we're a bit far of achieving food security at the moment. So it's important to increase our biodiversity, not only in our diets, but in agriculture, if we aim to battle malnutrition and hunger. We need to invest more in improving our underutilized crops. I would like to thank my supervisors, Sean Mays and John Bramwell, the technical team, as well as our collaborators in Malaysia, Professor Festo and Wei Kwan. And I would like to thank you for your attention.
Thank you, Nikki, for that call for the underutilized legumes. The last presenter for today is Natalia Leiva from Institute of Agriculture and Fisheries Research and Training, Spain. And she will be talking to us on genome-wide association study of drought-resistant related traits in Faba bean.
Good morning, everyone. My name is Natalia Gutierrez, and I'm going to present our work entitled Genome-wide Association Study of Drought-related Resistant Trait in Faba bean. Grain legumes are among the most important worldwide sources of high protein for food and feed, and key crops to support sustainable, low input and diverse farming system. In crop rotation, legumes enhance soil fertility through biological nitrogen fixation and break disease cycle, thus reducing the chemical use in agriculture. Faba bean is the sixth legume crop in production and is adapted to diverse climatic and soil conditions. Despite these advantages, faba bean still has a limited usage in agriculture, mainly due to their yield instability, explained by the effect of varied biotic and abiotic stresses, mainly drought. As a result of climate change, droughts are predicted to increase in both frequency and intensity, driving crop yields down. A meta-analysis of gene data along 34 yields in different legumes showed a gene reduction in faba bean of 40%, following to 65% reduction in water availability, revealing the highest drought-induced yield reduction when compared to other crops. So, the objective of the present study were evaluating the drought tolerance in 100 faba bean sessions using 10 morphological, phenological, and physiological traits. Conduct a genome-wide association study to identify single nucleotide polymorphic markers associated under stress, and investigate the relationship existing between the significant losses associated with the drought tolerance index. As a plant material, we used it on a panel of 100 Fabian sessions from different countries, evaluated at Germany under control and drought conditions during 2 years. The trial design was a randomized block with 4 replications in both the irrigation control and the drought stress condition performed under rain-out circumstances. The drip irrigation ranged between 60–70% of field capacity of soil, and the water contained in the soil was assessed by TDR probes. Drought stress was initiated when about 30% the plants started the flowering. A total of 10 phenological, morphological, and physiological traits were evaluated. So maturity date was the phenological trait, plant height, number of pod per plant, number of seed per plant, 100 seed weight, and plot yield were the morphological trait, and the physiological trait were free proline content, total content of soluble sugar, and chlorophyll content at the beginning of the stress and after 4 weeks of drought stress. Our traits were independently adjusted for field microenvironmental heterogeneity. Phenotypes were combined and adjusted by years using the best linear and unbiased predictor, and the method used was the following. A genotypic mean of the spatial adjustment phenotype was calculated for each trait and used for the GWAS, and all measurements were tested for deviation from normality by cuckoo plot. A correlation matrix was made by Pearson correlation analysis, and the heritability for the traits was estimated using this form. The genotypic mean value for each accession and each trait for the control and stress condition were represented by PCA plots. For genotyping, we used the Vicia Faba action SNP array with 50,000 SNP. After quality control, final matrix consisting of nearly 22,000 SNP with 93 accessions, and the imputation of the missing data was performed by mean allelic frequency. The genomic relationship matrix was calculated by first method of Van Raden. The genetic structure to estimate the number of distinct genetic clusters using fast structure, and the discriminant analysis of principal components. We use it to confirm the K number. A multi-locus mixed linear model was implemented to evaluate the trait SNP association. The standing Bayesian information criterion was used to determine the number of QTL. The Monferroni threshold was used to establish a significant SNP, and the significant marker were visualized with Manhattan plot and the p-value with cuckoo plot. Now I'm going to show you the result for this work. Here you can see the correlation matrix for the stress value using the Pearson correlation, morphological trait, and the chlorophyll content after 4 weeks under stress show the highest correlation values. A PCIV plot was performing between the 3 clusters detected under control and drought treatment, And as you can see, there was a clear separation between treatments. The heritability ranged between 0.29 to 0.73. 24 SNP were detected. No significant SNP association were observed for number of pot per plant, chlorophyll content, and total soluble sugar. The result for proline content, for example, identify 8 SNP and explaining 99% of the variation. Since the Faba reference genome is still not available to understand the genetic basis of drought stress, we blasted the sequence planking SNP associated against the Medicago truncatula reference genome. Several protein kinases were associated with plant height and proline content. This protein has shown to play an important role in drought tolerance in other species that Arabidosis, rice, and soja. In summary, our results contribute to a better understanding to the genetic architecture governing drought tolerance in Faba. 24 SNPs were significantly correlated with the straight study. The identification of the stress-inducible signal provided valuable information on the Faba response against drought stress, and a validation of the identified marker is in progress. Finally, here I show you the co-author of these works, and thanks for your attention.
Thank you, Natalia. And we had very fantastic presentations so far. For the next session on question and answers and conclusions by the ICLGG IAB, I would hand over to Judith. Over to you.
Yes, so Janila, I think we can you tell me how much time we have?
We can take, I mean, we are now actually 1:30 p.m. in India. That's what we said, 3 hours, but then we can take 15 minutes for this session. Yeah. Because of technical issues, we got delayed. Yeah.
Okay. So I can see that there is one question on the chat from Noel. Well, I cannot find it anymore, but
would you like me to ask it?
Yes, go on.
So it was a question for Jumping Wang about the that rather interesting study where there was a one-to-one segregation for the nodulation phenotype in the big B, little b heterozygotes. So, and the interpretation was that there was imprinting from I think the paternal allele. So obviously half of the big Bs are going to be paternal. But I just wondered if there was any background genetic analysis also done within the big B, little b heterozygotes to see if there is an effect of any other locus. Clearly, the one-to-one segregation is a very good explanation, but 9-to-7 ratios are quite difficult to tell from one-to-one, for example.
Yes, so is Jiang Hing present?
Ping Wong, she's from University of Georgia. I mean, I don't see her here because of the time differences we are running into across the globe.
Okay, so maybe the best would be to ask her by email.
Thank you, Judith. I'll do that.
Yes. So there is another question. Is there any role of specific microorganism in drought tolerance in chickpea? Maybe Kadambot can answer?
Professor Siddiq said he would leave at this time exactly.
Okay, so maybe for the questions we will deal this by emails.
Right.
Okay, so I just wanted to finish this small remote, well, not small in quality, but small in terms of time conference by trying to give a broader view of what we have been hearing this morning. At first, I would like to thank all the presenters for their very interesting talks. I think that the quality of the talks was really great, and I really thank the presenters. And during these talks, we have heard about different things, about the urgent questions that we have to tackle. We have heard about climate change, including drought, but there is also high temperature. There is also increased pest pressure that are important questions that we have to tackle. We have heard also about the need to develop the production of plant proteins for human food, including reducing anti-nutritional factors. We have heard about the different technologies that are not now available for us to tackle the important traits. High throughput phenotyping, we have seen very nice presentation about this. Also results about high throughput sequencing, high throughput genotyping tools. And I think that now, and the talk of Noel really gave an example of this. So we have plenty of data, we work hard, but we really have to work on the interpretation of this data, on the analysis of the data, but not only the analysis, the rough analysis of the data, but the interpretation of the data. And so I think that we could open maybe the discussion on this question, and if different people from that have been giving talks want to tell their vision on how we should proceed now in terms of genetics and genomics, I think it would be really interesting. I think that with big data, we need cooperation and cooperation among disciplines and among different groups around the world to give us the clues to breed quickly for adapted varieties. So maybe some of my colleagues may add their views to this few words of conclusion. Yes, Noel.
Yeah, thank you, Judith. I think your comment that we have to spend time trying to interpret the data is really important. It's very easy just to get lost in just in the excess of data without really trying to understand it. I thank you for your kind comments. I think it's very interesting that Mendel was concerned with peas because he thought they were the most nutritious crop. He was also beset by the problem with an insect pest, which caused him to stop his work on pea. And I think also we should bear in mind that a lot of his work was ignored for a long time, essentially ignored. And that's really to do with the second point that you made about being well connected with each other in functional international groups. That's, I think, really important to bring things forward.
Okay, thank you, Noel. Maybe does anyone wants to say something more? Well, Yes, Sean.
Sorry. Really, yeah, no, I mean, I think the point Noel's made is extremely important, is that it's so easy to generate data. It's still quite expensive to do it, but without a proper framework to interpret the data, we end up with things like vast numbers of candidate genes and no real idea of exactly what's happening. So the transdisciplinary approach is absolutely critical in terms of understanding, but also that needs to be plugged into breeding programs. I think that's the key thing. We need that translation to make impact. It's things have changed a lot in the last 20 years, and there is a lot more transdisciplinary work, and there's a lot more appreciation of the importance of doing it. So I think it's quite positive, I mean, in terms of where things are moving.
Thank you, Sean. Yes, Janila.
Yeah, so thank you, Judith. From where I come as a legume breeder, and the examples shown by Professor Siddiq, wherein the genetics of trait, understanding of the trait, of physiology coming in there, identifying the markers and introducing them into the elite background. I mean, the whole process, you know, I mean, this platform I see as a convergence of all the disciplinary teams coming together as we move forward towards having such kind of breeding varieties faster with better traits and all. So I see that. And in terms of, that's about the disciplinary. In terms of a trait per se, what excites me is the biological nitrogen fixation. How is it going to be different, better, or worse in the future climate change scenarios? So in the first instance, we didn't even understand how this processor in different legumes we have and how it's going to affect. And that's one area which would, set aside legumes. They are important and much more advantageous. So for the climate change scenario. So that's all my thoughts at this point of the time. Thank you.
Yes, Paolo. Paolo, yes, we would be happy to hear your comment. You are mute. Maybe is it possible to demute Paolo?
Please
at least I think you can hear me now. Can you?
Yes. Yes.
Yeah.
Good.
So I can tell three things, Judith, that your reasoning about interpreting data rose into me, let's say, you know. One, drought tolerance is very important to many of us. And I found very useful to analyze it partialing out, eliminating the effect of phenology. Phenology can have an overwhelming impact as a drought escape mechanism, but very often we can't exploit that so much. And it is kind of confusing what we could find, no? About other traits associated to intrinsic drought tolerance. And a second thought is we may study the genetic architecture of some physiological traits, but sometimes they might be the consequence rather than a cause of drought tolerance. And the third thought is studying the genetic architecture of traits. I mean, investigating QTLs can be very important to reveal that, especially for a complex trait such as drought tolerance or intrinsic drought tolerance, they are definitely polygenic, as we would expect. And this would automatically lead to give importance to approaches such as genomic prediction, genomic selection.
Yes, thank you very much, Paolo, for these comments. Very useful. I totally agree with the importance to dissect the traits, because most of the traits we are dealing with are complex traits. And yeah, so there is the impact of epistasis, there is also the impact of the environment, and it's, yes, very important to try to have the complex picture, to tackle the complex picture of traits. There is also one topic that emerged from the different talks, is that we have plenty of species among legumes, and so it's a lot of work, and maybe among the cooperation that we could increase is the work to make parallels among the different species. And also there is the question of underutilized crops, which is an important topic, and of wild relatives of our crops. Does anyone wants to comment on this? Okay, so maybe I think, Janila, you told us that we had 15 minutes, so just 1 minute left to thank again you, Janila, and the whole ICRISAT team that organized this meeting. And I think that in terms of cooperation, meeting in person is really important. So I invite you to come and join us in Brisbane in 2024. And please remember, if you're interested to be part of the International Advisory Board of ICLGG, write to me, and we will vote for the new advisory board at the Brisbane meeting. I leave the last word to you, Janila, and I thank all the people that are with us to listening to the end. Bye.
Yeah. Thanks, Judith. On my behalf, let me also thank the chair and also the International Advisory Board of ICLGG, and the host ICRISAT, the Director General Jackie Hughes, and the leadership team over here at ICRISAT and other teams, and also from Indian Council of Agricultural Research, Dr. Sanjeev Gupta, and today's honorary speaker, Dr. Noel Ellis. That was a fantastic talk, and all the speakers as well. Thank you, and bye-bye. See you in Brisbane.